Okay, well, hello everybody, and welcome to our latest Still Coveting chat. I'm Kara from the Hernandez Darling on Facebook, and today we are here to speak with my new friend Rick Byers. Rick is a engineer during the day, and he hacks testing at night. So we're really excited to hear about his setup, and everything that we could possibly maybe replicate. I don't know, we'll have to see. But thank you for being with us, Rick. We sure do appreciate you coming to share your experience and your knowledge with us.
Awesome. Well, thank you for having me, Kara. I love talking about viruses and molecular biology. It's a fun kind of passion project of mine. So always happy to chat with people who are interested.
We really appreciate the generous gift of your time. Now, we're going to do this the way that we normally do. Rick and I are going to talk a little bit about his testing setup, and then he'll have some slides to kind of share with us. And so please hold your questions until the end. You can type them in the chat, and then at the end, we will take Q&A. So without further ado, Rick, do you want to tell us a little bit about kind of your journey and how you got to having a testing? I mean, I called this testing in your garage. It's obviously, I don't know, is that it's in your
It's
garage,
in my
but
basement. Yeah, you can see it's just like a bench
yes, I see
behind
it's
me.
like right behind you.
Yep.
But how did you decide that you were going to like test yourself and your family at your house instead of, going out conventionally to get your regular PCRs?
So I would say it started, I took a little bit of genetics in university, but once I had kids, it was probably many of you have experienced, right? Kids are just germ factories, right? And I remember, when we first had our son, he was sick a lot. And then once he had, his breathing was sounding pretty bad. So we took him to to the doctor, and the doctor said, oh, his blood oxygen is low. Like, we're calling 911, rushing him to the hospital. And they said, well, he's got asthma induced by cold viruses. And they just kind of casually said, try to avoid avoid exposing him to people with colds. And we're like, yeah, right. Like, he's got to go to preschool. We've got friends, right?
Right.
And knowing a little bit about the molecular biology and being like a computer scientist and interested in kind of the information aspects, I just always pictured the viruses just always sitting there in the air. And I was a fan of Star Trek Next Generation as a kid. And so like I knew it was just a matter of time before we could really easily visualize all the RNA in the air that tells us exactly what viruses are there. And I just had this dream that surely we're going to be able to do this at home easily with cheap devices someday. Um,
And
and
this
so
was
at that,
pre-COVID. This was
yeah,
pre-COVID.
yeah. This was my son's 19 now, so this would have been like 17 years ago. I think I posted to the DIY bio forum then, asking for advice, and I just couldn't figure out how to make it practical. I was like, oh, this is going to cost me tens of thousands of dollars. How am I going to do it? And I would explore off and on every now and then, but I never really got serious about it until the pandemic hit. And then I was like, oh crap, I guess I should have taken this more seriously. And so like the first week of the pandemic, really, I was talking with some community folks about how we might be able to help our community, and I dropped 20 grand on a qPCR machine. And like I knew testing was going to be important. It was pretty clear very early on with some of the studies that there's asymptomatic spread and whatnot. So I felt not quite prepared, but also I spent the last 10 years thinking off and on about how we might be able to rapidly accelerate testing. And so at that point, I no longer cared about the practicality of, a few tens of thousands of dollars, and I just spent a lot of money to build it.
Right. And then I was looking through kind of the material that you sent over to me when we were first talking. And it looked like now there were some machines that weren't $20,000 that were like good enough to get
Exactly.
what like your common person would need to kind of
Yep.
do COVID testing or even, kind of viral assays. Is that
Yep. And
the
I'll show
case?
you once we put the slides up. We'll show you some pictures. But this guy here, I really like. This is called the mini PCR. I think it goes for about $600, $700 US or something. And it's just a thermocycler, which means all it is, you can put, this is a 16. So this is a bit more expensive one. Maybe this is $1,000, but there's an 8 one as well. And this one you can put 16 tubes in there, and it just heats it up and cools it down on the schedule that you give it. And so it's not fundamentally that complicated. The problem is actually getting a readout. Running PCR is just cycling temperature. It's no big deal. But to get a readout, you've got to do something with it. And so, I started with this $10,000 qPCR machine, which is how the big labs do it. But actually, let me just show you quickly. Grab
Sure.
my I didn't discover all this until later, but once I started buying things from Mini PCR, they had this really cool little box that all it is like a blue LED
Mhm.
uh, inside, and you put your tubes in here, and then there's an orange filter that filters out the blue. And so you can, same way a qPCR machine works, you get the fluorescence excited, and then you can see green, red, or orange colors in your tubes, and basically just eyeball to see
So it's an
did it
eyeball readout
amplify.
instead of like a paper readout.
Yeah, exactly. So you can
Yeah.
order this for $50 from Mini PCR. It's
Wow.
just some LEDs and a battery box basically, and an orange filter.
So I know that's the number one question on everybody's mind right now. Like, well, it's can we do this? But the number one question is like, what would be the upfront cost to get started? Say you were just interested in COVID. Now I know you do a lot more things, and I'm actually fascinated by the comparison of like which viral strain you have versus which viral strain your son has when you have a cold or something like that. But say, I mean, you just wanted to set up to do COVID testing. What do you think like it would be ballpark your outlay initial expense?
Yeah, I mean, the challenge really is probably in ordering the reagents and whatnot. I think you could probably get something working um, for a few $1,000s, right? If you just want to do COVID. Now, this isn't really going to give you anything better than I would say the Plus Life is better than this, right? In
Huh?
that, for example, you run this for a certain number of cycles and then you check to see if there's light there, right? And so
Right.
if you want to get a sense of how much COVID is there, you got to stop the PCR, check to see if there's light. Oh, there isn't now. Let me run it for 5 more cycles. Now see if it's light. It would be kind of labor intensive, and but you could do some basic PCR. Um, One of the challenges I mentioned in my slide deck is this stuff is not set up to be sold to consumers. It's designed to
Right.
be sold to research labs, but it depends. So mini PCR is great because they're designed to be sold to like high school teachers. So you can order
Uh-huh.
anything you want off mini PCR, but they don't sell the actual test kits for COVID. Those you've got to
Oh
order
yeah.
from a company that deals, and then so that makes it a little bit more complicated. You need a company that you can have them sent to, basically.
Yeah, that makes sense. I just, a lot of us are Plus Life users, or metrics users, like, NAT testing and that kind of stuff. But there's always this, kind of overboating fear about supply, right? Like, when will we be able to import? When will we not be able to import? Will it all of a sudden run out? Especially with all the stuff now too, with the conflict in the Middle East and all of the, how much, are we gonna is Europe gonna run out of jet fuel? And so then how are
Mm-hmm.
we going to
Yeah.
get our reagents and stuff over here? And so I think having, and now again, some of that's also going to affect the stuff that you're showing us for sure, right? Like some of the components and everything are still going to be sourced internationally, but having, kind of a backup for something more domestic, that there is some domestic supply for, even if it is a couple of $1,000s outlay, definitely I think is interesting to some people. And you just, let's clarify for everybody, you can't really like set up a home lab and go out there and advertise and say an all PCR test you, because eventually you're going to have CLIA and other regulatory agencies knocking on your door going, hey, what are you doing? You cannot be a public for service lab, even if you're giving it away without certain like regulatory things. But you can do this for your family. And if your friends happen to be over, you could do it for your friends, like you just couldn't advertise it or put it out there. So I could see, we all have pods, during COVID, and even now I say during COVID, during when everyone else thought there was COVID, we all know that there's still COVID, but we all have kind of pods or people that we could associate with, and I could see maybe some cost sharing and people kind of coming together with the outlay. Maybe it lives at one person's house, but friends and family can kind of, especially over the holidays or other times, where we really kind of want to gather with people and it makes it more difficult. So I think that's kind of what we're talking about here in terms of, is it feasible? Well, look, here's somebody who's done it. Okay, well, how much did it cost? What kind of background did you need? Because it's not, like you said, it's not easy. it's not like you put the card in the Plus Life and push the button, right? You've got to learn a process. You've got to source, reagents and machines and things from different places. You have to be able to kind of manage those pieces. But for those of us who do have maybe a science background or a healthcare background, that could kind of feasibly wrap our heads around. Like, I guess I want to say this is not too for somebody who, maybe doesn't have any kind of background in being able to do this stuff. It's not going to be as easy as a metrics or a Plus Life where it's like
Yeah.
in consumer, you can just do that. You are going to have to learn kind of processes that I'm hoping Rick is going to talk to us a little bit about today. But I do think even, it's not without, I know it's not without, not out of the range of what people would be willing to do to outlay a few $1,000s to have kind of continual access to something that they don't have to worry about it being jerked, because of imports or tariffs or fuel supply or anything like that. So that
Yeah, it's
being
a good.
said,
I don't know if my supply chain would be any more reliable. I'm always worried, that I used to not talk very openly about what I'm doing. It took me a while to really understand the biosafety regulations and whatnot. And I'm in Canada, so I've only looked at the Canadian regulations. I can't comment on the regulations in the U.S. But I eventually decided that I wasn't doing anything illegal, and that I
right.
could talk about what I'm doing. But still, a lot of companies aren't going to want to deal with me, right? Because it seems
Mhm.
unusual or whatever. A lot of companies do. It was really getting into the education side. Like I said, if you're interested in this stuff, even just starting from a curiosity of education, right?
Right.
There's some kits on mini PCR. You don't have to buy the full PCR machine. They've got some great educational kits starting at $100, $200. I had so much fun just getting my first pipetter. And
Right.
I never did any biology in school. Basically, I'd never done any wet lab. I did a little bit of genetics, but it was all like theoretical. I never touched a wet lab. I never touched a pipetter until the pandemic. And then I ordered a pipette kit from Mini PCR, and they have a course on how to learn to pipette carefully. And I don't know what it is. Something about my computer brain. I just found that super cool. Like, oh wow, look, 1 microliter of water. It's this tiny little bit of water, right? And and so if there's some if you I wouldn't recommend it for somebody that's just using the Plus Life and worried about supply chain, right? We gotta solve this problem another way. Obviously, we need to, get the market actually supplying these products. But if you actually care to play with this stuff and get your hands on it and experiment, then it's a great hobby.
Oh, but there's different driving factors too, right? You mentioned your son and his asthma and
Yeah,
that kind of stuff. I mean, I have immunocompromised kids, and so maybe I am a person who's kind of just worried about Plus Life chain, but at the same time, I
yeah,
have some real reasons, for doing that. And I homeschool my kids. I'd love to get a mini PCR kit and play with them
yeah,
and teach them because you don't ever know what you're gonna what that's gonna spark. And so
yeah.
I think there is kind of a wider or range, but I know that a lot of people here that is kind of one of the driving things is that they want to reliably be able to, I mean, and again, whenever you're doing this at home and you're doing this yourself, you have to understand your margin of error. It's going to be wider. It's going to
Yep.
be much wider than, even putting the card in your Plus Life or your metrics kit. And you got to kind of adjust your risk calculus around that. And you have to understand that it's going to take some time. So I think the driving home point is you can learn how to do it. It's going to take some time. It's not going to be something that you're just going to put together and push a button with. It might be something that you want to do as a group learning project with your friends, or with your kids' homeschool co-ops or something like that, so that everyone can kind of pool their knowledge and work together. It's not something that you're going to be able to get, and have done in a month and then have it reliably up and going and excited about. It's going to take time and effort and learning to kind of set up. But if you do, just the knowledge you gain, like you said, playing with the pieces of science and learning what's behind it, even if you don't end up at your kind of end goal to be able to test yourself for COVID or whatever, you're going to understand a lot more about what is behind the testing methods that you use. You're going to understand a lot more about what that Plus Life machine is doing or what your local PCR is doing and how those things, are putting together. So that in and of itself, especially for those of us who are bio-curious and also infectiously protecting ourselves, that gain may also be worth just the investigation, even if you're not going for the full-blown setup. So
One
definitely
other
all
piece that I would say relative to the Plus Life is I know there's been a lot of talk about expiration dates and trying to optimize, make sure you've got fresh tests, and sometimes they start to go invalid even before the expiry date. I've just been shocked at the extent to which I think using a lab setup like this, it's more predictable because you purify the samples, you get rid of all the gunk, and once they're purified, I think it's a more predictable, repeatable process. And for whatever reason, I've found my reagents, like they come with an expiry date of 6 months. I've used them 2 or 3 years after expiry, and I can't detect any loss in effectiveness. And so
Mhm.
basically for much of, except for sequencing, which maybe we'll get into, sequencing is a different story, but for doing PCR, I've been impressed that I just stock up. I order stuff once a year or something, and I just have a closet full of stuff, and I really just don't need to worry about expiry dates. Now, like you said, this is not medical advice. This is like, professional researchers would say, what, you use something after expiry date, you're crazy. But as we all know, this is all a bit of probabilities game, right? Like we're all trying to optimize, do you want to test? You're not going to test every hour, right? You're choosing how often you're going to test. You're playing a probabilities game. And so in my experience, I have not found older reagents to make a meaningful difference
Mhm.
than fresh reagents, which is a nice
Right.
change because I've definitely seen the problem with Plus Life where,
Right.
old
Yes.
tests are.
And just to clarify for everybody, Rick is not saying use expired plus lives. Please do not use
Yeah,
expired plus lives. We know, but there's a lag between the time they're manufactured and they sit in the warehouse, and they go to the distributor, and then they sit there longer, and then they have to be sold, and there's all other sorts of variables there. But if you're used to using them yourself, then and you can kind of tell what's going on. It's almost like if you're baking the bread yourself, when it's done. You can test it with a toothpick. You're used to it. Whereas if you just buy it from the store, you're kind of taking their word for when the bread is done or not done. So don't play with plus life's expiration dates, but it's great to know that if you're sourcing, buying, and storing your own, you have a kind of a wider variability for how long those reagents are going to last. And again, you're not doing this for the public, you're doing it for yourself, understanding your own kind of risk benefit analysis. So
exactly.
that's great information. So do you want to just kind of get into the nuts and bolts with your
Sure. Yeah.
presentation and kind of show people like
Yeah.
what
Sure.
you did and exactly kind of how you built that out?
Sure. Well, we'll walk through some slides here. We can talk as we do it. I originally gave a presentation back in 2023. I just did a few updates today just to correct some of the stuff that was really far out of date. But I kind of start by saying I often talk to software people, right? So I won't dwell on this part, but like if you read the history of software back in the 70s, computing was incredibly hard to access, right? And like the head of DEC was like, there'd be no reason for anybody to have a computer at home, right? And there were enthusiasts, there were hobbyists who like wanted to get these kits. They were really rare, and you had to kind of assemble them yourself and solder them together and whatnot. But it was kind of foreshadowing what was going to come to all of us, 10, 20 years later, when folks, who are really curious about it were trying to assemble, computers in their garage, basically. And so I see a lot of parallels between that history and what I think is happening now in healthcare and biotechnology. That's just a picture of my lab bench at one point years ago. Some of the things on there. This is me with my son Oliver, when, like I said, when he was 1 or something like that. And I told you the story already of him having asthma. There's him with his asthma puffer,
Yep.
trying to teach a 1-year-old how to do asthma puffers. But that's what got me started. And I wish I could go back, and I wish I had samples from back then because I'd be so curious to see, what were all the viruses going through our household at the time. But like I said, it took me 15 years or so to get to the point that I actually could. And like I said, I grew up in Star Trek, and so like I knew like Star Trek was great at predicting the future in a lot of ways, and I knew it was just gonna be a matter of time before there's no fundamental physics reason or biology reason why deep studying of the viruses around us is more complicated than say a laser printer, right? There's
Right.
microfluidics, there's like chips. Chips are incredibly complicated, molecular grade things that we have made incredibly cheap because of their volume, right? So if someday the real barrier right now, I'd just say, is volume, right? If everyone in the world wanted to test viruses at home, it would be cheap. You could have probably home sequencing, and it would be completely cheap. Oh, I first did this deck when AI image generation was just coming out. So this was my attempt to visualize viruses floating around my kid's room, that I couldn't see but really wanted to. Um, anyway, so do you want me to talk a little bit about the science, like actually what's going on
I
in the
think
test?
you should just give kind of a basic primer for people who may or may not actually be even familiar with
Sure.
with PCR and how it works. We have kind of the gamut here, I think, today, and that will watch us. some people will have been lab scientists and will have
Yep,
actually probably done some PCR even or played with it or kind of danced around it. We'll have other
yep.
people that are just consumers right now and they don't necessarily understand exactly what the basics of PCR are. So if you want to give us like a
Yep.
little bit of a primer, no pun intended,
Yeah, sure, will do. Yeah,
and
I'll
then
keep it brief and we can get into more in Q&A if people are interested. But basically, PCR is just a way of, I think of it as Control F, find, so that's these primers, these purple things here are, you design some sequence that says, I'm interested in this pattern. So that's what you're typing in, Control F, I'm typing in ACGGGC or whatever. And then Control C, Control V, copy paste, right? So I want to find anything that starts with this little sequence and ends with this other sequence, and then I want to copy it, and I want to copy it over and over again. And every time you run the thermal cycler, you heat it up and cool it down, it doubles the number of copies of things matching those primers. And the reality I've learned is much more complex than that. Everything in life, everything in biology, I would say, is there's exceptions and it's fuzzy and it's all probabilistic. But that's like the high level idea, is that you can have a huge amount of variety of DNA in a tube, and if you just say find the stuff that starts with this and ends with this, you can go and amplify that up. Like I said, it's probabilistic. It does sometimes amplify other things as well, and then that's where the challenges comes from. Building a sensitive test. But anyway, that's the theory. And so the first big challenge for me was like how to do this without spending a fortune. When I first started looking at it, I thought it was going to be $50,000 or more. Until I learned about mini PCR. And I don't know why I didn't find them. They've existed for a long time, but somehow I missed that they existed. And once I realized there's these stuff designed kind of high school biology lab grade stuff, I realized it was way more accessible than I realized it was. So there's a bunch of great stuff on their kit. I mentioned, this mini PCR device. Now, okay, another big challenge was, okay, it's possible to do PCR now. How do I actually choose what primers to use? Now, if you're doing COVID, it's easy. WHO and CDC have a standard set of COVID primers. They're published publicly. You can just order the predefined CDC and WHO ones. But I wanted to do a lot more than just COVID. And so I spent a lot of time reading different academic papers and finding different assays that people had built. This was one that was for RSV, rhinovirus, and metapneumovirus that I've used a lot in a lot of my tests. And so I just read a bunch of different papers, learned from what people had done. And part of the interesting challenge here, by the way, is rhinovirus is the most common cause of the cold. And so, I really like the feeling of being able to know what is it that I have, not just, I'm sick and it's not COVID. I really want to know what is it. And there's antigen tests for lots of types of viruses, but there's no antigen test for rhinovirus as far as I can tell. And I think that's because rhinovirus is incredibly diverse. There's like 70 different species or 70 different subtypes of rhinovirus, 130, I think there's 70 of rhinovirus A. There's 3 different species, rhinovirus A, B, and C. And then between all 3 species, there's something like 140, 150 different subtypes. And I think you would probably need an antigen test for many of them to have a reliable antigen test. But the cool part is there's a part of DNA in the rhinovirus that's non-coding, that doesn't change, that's conserved across all the different strains. And so if you're doing a molecular test instead of an antigen test, you can target that. And that's, but still, it was quite a challenge for me. I still found I have 3 or 4 different assays for rhinovirus, and I often will have a cold that 3 of the assays will hit on and 1 will miss it, because there's so much variation. So yeah, this is the 5' untranslated region at the end of the rhinovirus genome is where most of the assays target, because that tends to be conserved. Well, this is then you got to think, how are you actually going to get these primers manufactured? They're just a string of DNA letters, basically, and you can get DNA synthesis machines. This is kind of a low-cost one, but I think it's still tens of thousands of dollars. The good news is there's great websites. Basically, ordering DNA is as simple as going to a website like IDT, typing in the DNA you want, and hitting buy. And you'll see here, this was the rhinovirus forward primer, cost me $7.98 to basically get as much of it as I'll ever need. And you basically, they effectively print it for you and ship it, to your legitimate business in a week or so. Next big challenge, like I mentioned earlier, there is a real challenge of buying supplies. BioBasic is one company I use, but most of these lab supply companies have terms like this, right? They don't ship to residential addresses; they ship to research organizations. But there's tons of research organizations around, and you can probably find friendly research organizations. There are biohacking maker spaces in a lot of big cities that qualify, and so as long as you can find some legitimate organization that is open to doing a little bit of research, then you can get stuff shipped from these kind of companies. All right. Oh yeah, this was just me mentioning. This reminded me a lot of this homebrew computer club back in the days. Where Steve Jobs and Steve Wozniak. I think this is maybe one of the first Apple devices. People were building these chip like home computers in their garage. And to me, this is what it kind of feels like. You kind of have to. Back then, people talk about, if you read the history of people like Steve Wozniak, he would talk about having to go and beg and borrow people at the electronics companies for chips, because chips weren't something you could just buy, right? They were something, they were new and they were hard to get. So I think that's kind of where we are with some of this biology stuff. This is just a set of the different primers that I got a couple years in. You can see there's a bunch of common viruses in here. So like CoV is not SARS-CoV-2. That's trying to check for a bunch of different coronaviruses. But there's adenovirus in there as common source of the cold. RSV, respiratory syncytial virus, is also a common virus. All right, but then once you've amplified something up, now you've got the challenge of how do you see what's in there, right? Just because you have a lot of the DNA doesn't get you any closer to knowing, did you actually find what you were looking for? And so the most simple way to do this is a process called gel electrophoresis, which is you can make DNA glow pretty easily, which is neat. It's not that the DNA glows, but there are proteins that will glow in the presence of DNA. And a gel lets you separate the DNA by how big it is. So you can put an electrical charge on, it's basically a sugar. It's basically like Jell-O. And when you put your DNA in a hole in the end of the Jell-O and you put an electrical charge through it, it pulls the DNA through it since the DNA is negatively charged, and the smaller pieces of DNA can move faster because they have less kind of navigating the holes in the Jell-O. And so it spreads out the DNA by its length. And this is kind of the standard, molecular biology technique, I think dating back to the 70s, I think, for visualizing, hey, did I find what I was looking for? It just tells you how long it is. But if you know, hey, I'm looking for this sequence that's 500 base pairs long, and then you run your PCR and then you look at it on a gel, if there's a bright line around 500, you can be pretty confident you found what you're looking for. And this is an image of what a gel might look like here. You always use a ladder on one side, which is a standard set of lengths of DNA that kind of acts like your measuring stick for all of your samples. So now, right. So here's a gel that I ran, and you can see in practice, when I was looking at the papers, one of the problems that I find with academic science is scientists publish their successes, and you learn from your failures, not from your successes, right? And so, like, starting by reading papers and reading my biology textbooks, I thought this stuff was just going to work because, like, all the pictures look perfect like this, and then when I did it, man, it was messy. Excuse me, right? So this is one of the first gels I did, and there's ladders on either side here. You can tell even my ladders are kind of fuzzy, and then my samples are kind of all over the place. And ultimately, I attribute this to, like, PCR is not, there's a lot of effort that goes into trying to tune your PCR conditions to really focus on the thing that you want to get. And in practice, PCR is just probabilistic, and there's so much DNA in a sample. You've got 6 billion base pairs just of your genome, never mind the viruses and bacteria in you. And so these primers, they bind to other things all the time. They bind to each other, a process called primer dimers, and it creates all this junk that you kind of have to figure out how to ignore when you're doing PCR. And so the way professional labs, if you actually go get a PCR test somewhere, and I believe the way that Plus Life deals with this is they use a second step called a TACMAN probe, which is, in addition to the two primers, it's a third region of DNA that when it finds a match, it glows a certain color. And so you can use the PCR to amplify lots of DNA that looks kind of like what you're looking for, and then use the probe to say, now did I really find what I'm looking for? And this is a bit tricky because it can trick you into thinking, if your PCR is not very specific and it amplifies a whole bunch of other stuff, now there's a lot of junk in there that the probes aren't going to be able to find what they're looking for, and you'll get a negative instead of getting instead of getting like an inconclusive or something like that. So you're kind of trading your uh, your specificity. You want to improve the specificity so you can precisely find COVID, but you're kind of losing sensitivity when you do that. Now, there's a risk that PCR, when it finds other things, is just going to mask what might be otherwise a true positive. But anyway, this is how most commercial PCR testing is done. It's called qPCR, quantified PCR, specifically using these probes. Like I mentioned, you can. So if you can get qPCR probes, now that you can visualize them in a box like this, and in there, I think I just have a red one and 3 green ones. My workhorse has been this device. BioMeme is this company. They're super cool, but this is basically where I started, and out of desperation. But the BioMeme Franklin I'm still using the same device I bought March of 2020, and it's a qPCR machine. So it not only does the thermal cycle, but it has a little image sensor, and it's every cycle it's measuring how much red, green, and orange is it seeing in the tube. And then from that, I can plot these nice little charts. So concretely, this is called a dilution series, where I think this one wasn't a virus. I think this was a human gene. I use a particular human gene called beta-2 microglobulin as a kind of reference standard for myself. And one of the things you do when you're testing your PCR setup is you dilute your sample a certain number of times, and then you make sure that you actually see the cycle. If I dilute it by half, I should see the number of cycles go up by 1. So in this case, I think I diluted these by 16 times every time, right? So I would take 1 milliliter of my sample and add 15 milliliters of water. And then I would again do 1 milliliter, or scale that up. I probably was operating with larger volumes than that, but you have the idea. And when that works correctly, you get a plot that looks like this. Each sample, because it's 16 times dilution, that means it should happen 4 cycles later, 2 to the 4th, at each stage of your dilution. So this is one way you measure the efficiency and effectiveness of your PCR, doing one of these dilution series. And to me, it always boggles my mind every time I do a dilution series, and I'm taking the tiniest bit of sample. I'm taking 1 microliter, and I'm putting it in a bunch of water, and I'm doing it over and over and over again, and I can still find what I'm looking for. And it just makes me the scale of how tiny the DNA is, and what it means for there to be trillions of copies of a virus in a couple milliliters of sample. It just always blows my mind.
Exactly.
Yeah. All right, a couple more challenges I'll go through, and then we can go into more Q&A. A big one, you've probably heard Leo and others talk about it on the Facebook group if you follow that. But when you're messing with this stuff, when you're messing with amplification, contamination becomes a big problem. So pretty early on, I wasn't sure that my yellow channel was working, and so I ordered a synthetic control for my human metapneumovirus assay. I wanted to make sure, because I didn't have anybody, I wasn't able to get viruses anywhere. So I ordered something that would cause my test to go positive. And I don't know if I screwed up the math or what, but I ran it in my PCR, and sure enough, I got a strong positive. But I amplified so much of the stuff that it just infected the air. The DNA must have just been floating in the air in my lab. And from then on to this day, 5 years later, every time I test for metapneumovirus, it's positive. Because all of my reagents, all my liquids, I think, in software we make off by 1 errors. I think in biology it's possible to make an off by 1,000,000 error. And I think it was just I got a lot higher concentration of this control from the supplier I ordered it from than I was expecting. And so when I amplified it, it was just trillions and trillions of copies. So yeah, it's a bit of a problem. But even outside of this, I've had problems where, especially if you start doing gels, you're handling the amplified DNA, it's just really easy for it to get into the air into little droplets and float around and land in your reagents and contaminate things. So I try to separate. I've tried to get better at this. I have a bathroom off my office that I use for my post-PCR. I try to put a air filter in there and whatnot, but still, I'm paranoid about contaminating my reagents.
Well, and this is something that people really need to be aware of when you're doing something like this versus something like a Metrics or a Plus Life, because you'll hear Leo and other scientists kind of screaming about contamination and how it's like glitter, like how you can never get glitter out of your house. And I used to kind of laugh because when you're using those kind of more direct for consumer things, it's a lot less likely that you're actually going to contaminate stuff. But when you're playing with pieces of things like you guys do, then yes, then you can. I mean, they can have to decontaminate a whole lab based on one kind of contamination cycle. And so it is worth pointing out that you do have to be a lot more careful when you're doing it this way than you necessarily would have to do in just your regular old, whatever you're used to kinds of NAT machines they're selling. That's one of the reasons why regulations are so finicky for those machines, is because they want to make sure that, just everybody, average, Jane or Joe can't screw it up like this, because it's kind of easy when you're playing with the pieces to screw something up like this. And so, talking to your friends who are scientists, talking to people like Rick who've done it before, talking to some of the other members, because we have several members of our community that are very generous with their time that do PCR on the regular. And those kind of resources are going to be real important if you do decide to do an endeavor like, okay, I'm going to try to do this at home, but now it's messing up. It's not necessarily saying give up if you mess up. It's saying expect to mess up, a plan for messing up, and then see who's your phone a friend, or how are you going to kind of get back on track once you do.
Yep. Yeah, exactly. All right. Oh, and I should say, with Plus Life, as long as you don't open the top, like definitely when you're done running your Plus Life, do not open the top and shake the liquid out. That would be a bad idea. Other than that, I don't think you're going to have to worry about contamination. But yeah, if you're running a gel, like you're taking some of the liquid out and you're putting it in and you're leaving it kind of exposed to the air for a while. So contamination is pretty easy. The other challenge that I had I finally built a PCR lab during COVID, and my family was really good about not exposing ourselves, and so nobody was sick anymore. It was the first time in my life that we went, I think, 2 years and nobody had a cold. I was like, hey, I finally have this lab, yay! But I have nothing to test it on. Now, luckily, at the same time, I also got busy we launched a volunteer program. This was before rapid tests were widely used. We launched a program in our community where we had these buses. The only legal way to be able to use rapid tests at the time was to have trained healthcare professionals. So a bunch of us figured out how to become officially trained healthcare professionals. We got the government to buy a bunch of tests, and we had these buses that went around where we were doing rapid tests on folks. That's another story, but it was neat, fun experience in the community, trying to help the community. And it really was, the rapid tests were obviously way more scalable and deployable solution than what I originally thought. We were going to try to deploy these PCR machines at scale, and it was just, the expense, it was just, not practical. All right, and then finally, I think maybe a year and a half in or something like that, I had a run where I have a positive control to make sure my swab is good. There's my contamination that's ever present, but I finally had, I had all these negatives, and I finally had my first rhinovirus hit. And I was so excited that finally I was able to detect a real virus in my lab.
And you have to qualify all your results to everybody to show them your contamination every time. That's awesome.
Yep. And then immediately started thinking, well, now now I have rhinovirus. Now I want to know what rhinovirus it is. Is it A, B, or C, and whatnot? So now I want to sequence it. I want to actually get the DNA letters out. And I knew sequencing was getting cheaper and cheaper. I think I'd already bought my full genome sequence at that point from a company. So I figured it was going to get easier and easier. But I couldn't find any service that would do it for me. And so I eventually, there's this neat company called Oxford Nanopore, whose mission is to enable the analysis of anything by anyone anywhere. Really resonated with me. And I finally managed to buy a nanopore sequencer. And these things are amazing. They're here. I've got it right here. And this is the important stuff is in here in the flow cell. And basically inside the flow cell, there's these genetically engineered pores that basically there's a motor. They've engineered this motor that pulls the DNA through the pore. And as the pore gets bigger and smaller, as the DNA moves through it, it creates an electric current in this chip, basically. And then there's an AI system that converts those electric currents into a series of ACs, Ts, and Gs. So it's basically reading individual molecules at the molecular level and converting it into software signals. And it has a high error rate, but it's fine because you just run it enough that the errors can cancel each other out, basically. And so for the main flow cell, the MinION, costs about $500 for a flow cell. It's really finicky on lifetime. They last only 12 weeks. I think I had some that didn't even quite last that long. And then there's about another $1,000 of reagents involved and a lot of hands-on time. I was getting to the point that I was only doing sequencing once or twice a year because there was enough work and expense involved that I would save things up and run a sequencing run. I took a week off work once to go do a backlog of all my sequencing. And then this is kind of the result that you get. This is at the bottom is a reference of a rhinovirus genome, and each line is one of the reads. Where it's gray, it just means it matches the reference, but where it's a color, it means it found some difference from the reference. And you see in here, there's a lot of noise. That's just the errors from the sequencing. It's imperfect. But then you see some places like this C here, the C, the A, and this C, where there's clearly mutations in the rhinovirus I had relative to the reference genome. And those patterns of mutations basically tell you exactly what strain you had and let you answer questions like, do I have the exact same virus? Did I get it from my son? Did I get it from this person? Or is it different? And with rhinovirus, you get about, I think it's like one mutation every 1 or 2 transmissions or something. So you can effectively reconstruct a full family tree of rhinoviruses if you have their full genome sequence. And so the first thing I ran this on, of course, we had COVID in, was it 2022, I first had COVID. And it was the first time we decided we're finally going to travel again. And when you know it, like within 2 days of starting our trip, we got COVID. So great. But I got to sequence it. And I was so excited to be able to say, this is the COVID I had, BA52128. This is the family tree of COVID at the time. And this is exactly where the one my son and I had. Another challenge is just this stuff is all messy, right? Molecular biology, it's so probabilistic. Things are never as clean as you expect. You want to get these nice S curves. I get, different shaped curves all the time. I don't quite understand why. I had this one that was really driving me nuts, that my rhinovirus assay was not working very well and I couldn't figure out why. And then I finally sequenced it, and my goodness, this is my primer here. My primer goes right at this region, ends in a C, and this rhinovirus happened to have a mutation, a T, right at the most sensitive part of the primer. So because it wasn't quite matching the most sensitive, it was a 1 base mismatch at the most sensitive part of the primer, it completely explained why I was getting these really unpredictable, unreliable PCR results. And so this is kind of the power of sequencing, to be able to really debug and understand. As a computer guy, I think of it as this is kind of your debugger, and you can go in and poke at the memory and see what's there. And so this is just one analysis I did. I've got a bunch. If you're into this stuff, you can look at my GitHub, Byers on GitHub. I've got a bunch of different analysis that I do. But this was just looking across the different rhinoviruses I had, how well did my different assays match to them? And I could see this one assay I was using, HRV-MA, is a paper published by some guy whose name ended in started with MA. Wasn't matching very reliably to the rhinoviruses I was finding in my house. So that helped me convince me to switch to some of these other assays.
But don't feel like you have to sequence rhinoviruses if that's not what you're here for. That's
No,
for those of us who are, kind of molecular geeks.
Yep.
If you're here for COVID assays, you can, once you figure out that it's not COVID, you don't have to go and figure out what it is unless you just want to. Or you might do your sequencing part to figure out that it's not COVID, and then you might resort back to, your flu and RSV rapid test. And then if that doesn't work, you can move on from there. So don't feel like if you're trying to explore this, I know some people listening might be overwhelmed by thinking, oh my God, I've got to do all of these extra things and all that. You don't. You don't really. You can start very simply. You don't have to figure out which cold strain that someone in your house has. You could just start with yes, COVID, not COVID, and then move on from there as your, knowledge and expertise expands.
Yeah, exactly. Like getting into PCR is one step. There's some complexity, there's a learning curve, and sequencing is kind of a whole nother step up. I've now gotten to the point, by the way, I don't do sequencing at home anymore. There's great services now. There's one called Plasmidsaurus. I can just ship my sequences off to them. It's like $20 a sample. It's like way cheaper and way faster than I could do myself. But yeah, still, if you're curious about it and you want to dig into the details. But it does sometimes come in handy. And so this is one example where we're always trying to adjust how we isolate from each other in the house when someone's sick, and learn about what works and what doesn't work. So there was once my son had a cold, and we tried to avoid catching it, and then 17 days later I had a cold, and so I posted to Twitter, do you think I caught his cold or not? And actually I didn't. This one I was able to figure out without having to sequence it. There's another analysis you can do called a melt analysis, where once you've done your PCR, you check to see how hot do you have to get in order to separate the DNA into its 2 strands. And in this case, all of the ones on the right were all different samples from my cold, and the one on my left was my son's cold. So I could tell they had to be a different sequence because they had a very different, the amplicons had a very different melting temperature. So I was able to say conclusively, nope, it's not that our isolation failed. It's that I just happened to get a rhinovirus a couple of weeks after my son did, but it was a completely different one. Few things that I continue to struggle with. I just took a new photo of this because my freezer, I keep having to figure out what to throw out of my freezer because I'm not willing to buy a gigantic freezer. So my freezer is jam-packed full and I have to decide what to get rid of. There's still a really big question of, obviously people vary, but everybody's got to decide for themselves. How do I actually want to change my behavior based on what I'm doing? So when I've got a cold, I don't go to work, for example. But one thing I've learned is, especially with rhinovirus, I'm probably contagious a day or two after I feel like I'm better. Like my runny nose is cleared up, but I'm still just like slightly congested. And this is a graph here showing the viral load over time. And I was shocked to see that the viral load near the very end of my cold, where I just have a little bit of congestion, is basically as high as it was throughout the whole progression of the cold. And this is a log scale. So, this is PCR cycle count. So every number up is twice as much virus in there. So, this is right up until, say, day 9 here. I'm certainly contagious, maybe on day 10 as well. So that's definitely changed my behavior. When I'm sick, I now wait till I'm PCR negative before I go back to hanging out with my family and going to work and whatnot. And then I still struggle to some extent with being able to detect everything. There's lots of challenges. There's all sorts of reasons why things might fail. I'm now up to about, since 2023, in total, I've sampled about 44 different colds for my family, and I've been able to confirm 80% of them PCR as being something. If I exclude before 2023, where I didn't have it all figured out, I'm up to about 86%. And I've sequenced some portion of at least 21 of those. And you can see the viruses over to the right. This is just what's gone through my house, mostly rhinovirus, as I said. That's the HRVs there. Seen flu A and a little bit of flu B in our house. 7 cases of COVID have gone through my house since I started doing this. And I just had RSV-A myself a couple weeks ago. It was nasty. I didn't like it at all. And then a few other random viruses here and there. Including I had shingles at some point, not a respiratory virus at all, but that's the VZV, varicella zoster virus.
Yeah.
And then it's just like there's a real cost of entry, and this stuff is not easy. I've probably spent thousands of hours at this point experimenting and learning, but I have fun with the failures. I learn from the failures. This was just labeling, some of the stuff in my lab.
Mm-hmm. Well, let's back up just for a second.
Yeah.
And so again, thinking about people who are just kind of, bio-curious and COVID cautious and they want to get into this. You're talking about, the mini PCR, right? And starting with the mini PCR and maybe getting some of those high school mini PCR lab kits, right?
Yep.
What other, if you just want to do that, I have COVID or I don't have COVID. That's all you want to
Yep.
do. That's all you want to learn how to do.
Yep.
And you're going to use the mini PCR kind of setup to segue you into that. What are the things that you need?
The first thing you've got to do is you've got to purify the sample, right?
Mhm.
The Plus Life and others use crude samples, but it's really unreliable. Any PCR system is really going to want a purified sample, just the nucleic acids, the RNA. So let me just grab for a second. I prefer, you got a few options, but I use these from a company called BioBasic, and they're these tubes where you put your crude sample in, and through a series of washes, you're basically collecting the DNA onto a little membrane. And so in order to do this, it's called a, sorry, I'm forgetting to name them. Anyway, flow something or other. Anyway, in order to do this, you need a centrifuge. So you got to be able to spin it and create some. So there, you see on the left there is my centrifuge. I think that's $400 for mini PCR or something like that. There are other options. The company that I bought my qPCR machine, what I started with was a kit that used the same idea, but used a plunger syringe instead of a centrifuge. And that's great. It's just those purification kits, I think, were, $30 a sample, $40 a sample, or something like those. They're called the BioMeme M1 kits. So they're a great way to get started if you just want to do a few. But 30 or $40 a sample was too rich even for my blood. So these, I think, work out $2 or $3 a sample or something like that.
And what are those
So
called again? You said those are from which company and what are they called?
So the cheaper ones, these ones here,
Hmm.
yeah, they're called BioBasic EZ10 virus purification kits.
Okay.
And so you get, basically the process is you take your swab and you put it in almost like a soap that breaks the cells apart. So it comes with this uh, they call it a lysis buffer. So to lyse the cells. And so you sit it in that for like 10 minutes just to break all the
Hmm.
cells apart. And then you run that through one of these tubes, and it traps the material onto this membrane. And then you have to run a wash through it, a protein wash that washes a lot of the protein junk out.
Hmm.
And then you can,
And
once you've done that, you
the
dry it.
lysis buffer and the wash are with the little kits that it all
Yeah,
comes together. So
yeah, exactly. It's easy.
you have to have your swabs. So if you're making kind of a mental list in your head of what you have to have your swabs, and then you can get these little kits, and the lysis buffer and the wash and the little tubes are all together in this little kit, which is $2 to $3 a sample.
Yep, exactly. And
Okay.
then you need a centrifuge to run them on, and then you need micro pipettes. You need pipettes in order to be able to, move the liquids around. I just order pipettes from Amazon. That was actually it was a pretty easy part. You can get three different sizes of pipetters for $30 or something from Amazon. Cheap Chinese stuff, but it actually has worked. I've checked their volumes multiple times. They've worked great for me. You need to order pipette tips, $5 a box or something for a box of 96 tips or something like that. Some basic things like, you're going to want rubber gloves, probably want to wear goggles to protect your eyes, some cleaning, but you see I've got some wipes there that are just like, your normal Lysol wipes or something that you can use to clean up.
Mhm.
Um, you want some kind of workspace. So I have this blue tube rack, and it's just like a piece of plastic that you can sit your tubes in while you're working.
Mhm.
So stuff like that. It's kind of crazy how you can find stuff on Amazon or Etsy or something for $20, or you can go to the professional lab supply company and get basically the same thing for $500.
Right.
There's like this massive markup. If you're serious, you pay a huge amount, but you get basically the same thing.
Mhm.
You know, yeah. So that and then you just need the PCR machine and some way to visualize it. So
Mhm.
like I said, the
So
box.
you could get started with the centrifuge and
Yeah.
micropipettes and your your swabs, your tubes right there with your purification kits, and then you need your mini PCR machine and your mini PCR viewer.
Yep. And then there's a few reagents you need. So we
Mhm.
talked about the sample purification reagents, but then the next step is something called reverse transcription, where you convert, COVID is an RNA virus. So you got to convert that RNA to DNA.
Right.
So you can order a tube of reverse transcriptase enzyme. I use a mix from a company called NEB, New England Biolabs. It's fairly, I pay about $500 every time I get a tube of that. But I forget how much long that lasts. I usually only get a tube once every couple of years or something like that.
Mhm.
Um, I do have, I'll share it after this. I do have a spreadsheet that I made when I was first getting into this, comparing the per test costs.
Right.
I went through and tried to optimize all these different parameters. Like there's all these different reverse transcription enzymes. Which one am I going to use? All these different sample purification kits. And so I went and optimized a bunch of them to try to, I think I got my per test cost down to about $5 a test or something like that, factoring in the reverse transcription, the sample purification, the swab, and then when you actually do the PCR, you need what's called a master mix. You need a reagent that you actually put the primers and the sample in with, uh, and then the assay itself. So you have to actually buy the SARS-CoV-2 assay, which it
Right.
might be about $100 or something like that to get a small amount of it. I think
So,
that's about it in terms of the reagents. And then basically all I had to say after that is I'm confident this is going to get easier. So when I started this, home PCR was not a thing.
right.
And then the Q tests, and there was a few tests that came out, but the Q test was the first that I thought was a real exciting consumer one. I did a tear down and figured out how the Q tests work. And then there's these microfluidics lab devices where people are figuring out how to move small amounts of volume around without having to pipette, basically using magnets and microfluidic channels and whatnot. So this is basically a programmable lab for $7,000. I haven't bought this yet, but it's made by the same people who make the nanopore sequencer, and I'm excited to try one at some point, just to stop spending so much time myself just pumping liquid from one place to another. And then if you look a little bit further forward, the company Oxford Nanopore that makes the sequencer, they envision a time when it could be cheap and practical enough to basically just build sequencers effectively into your toothbrush. They've I mean, that's
Wow.
looking a bit far out, but this is from a presentation they had several years ago now, where they said they're increasingly automating more and more of the process, including the sample purification, the reverse transcription, the PCR, and then the sequencing. And it's not outside the realm of possibility that we could get to a point where it's not that expensive, that at some point in the future, every time you brush your teeth, there's something in there that is running this whole process and just spitting out some sequence for some software to analyze, to say, oh, here's the bacteria and viruses we found in your mouth today. And so that's
Wouldn't
kind of
that
the
be
future I
cool?
envision. Yeah.
Yeah, that would be awesome.
Yep.
I wonder
Exactly.
where they are with that. Have you checked since 20, I said this says 2022. Have you
Yeah.
checked to
Yeah.
see if they,
I watch their annual conference every year, and they keep getting a little bit closer. They're still pretty far away, I think, from the toothbrush. A lot of it, there's a scale problem here, right? Like, you know, Oxford Nanopore has done. COVID really helped create a lot more demand for this kind of stuff, but it's still nowhere near the kind of demand it would take to make it cheap for
right.
consumers. Right? And so they're still, and they're not really at the stage where they've perfected this whole, like I said, in my lab, I'm still manually moving liquids around with pipetters, right? So at some point, I hope to be able to get one of these like automated, big professional labs use big robotic systems, like $100,000 liquid handling robots, basically,
Yes.
but it's basically just a robot that's doing what the human's doing.
Hmm.
Where I think Nanopore is exciting because they're talking about how do we really miniaturize this and scale it, and you, instead of using big pipetters, using microfluidics, similar actually to the Plus Life, right? I'd say the
Right.
Plus Life is very much an application of microfluidics, and there's no good reason. I could imagine the Plus Life scaling. It doesn't have to just be the 8 channels that are in there. It could be 256 channels. Make them even smaller, test for a wide variety of viruses and whatnot. So I think the technology is there. It's just about the demand and the economics.
That
And
makes
that's all I
sense.
had in my slides. I ended with this one just because uh, these were my daughter's goldfish, or fish. Sorry, not goldfish. They're fish, but they're genetically engineered to glow different colors, which is apparently just a thing now at the pet store. You can just go get fish that have been engineered to glow different colors, and it's kind of funny how 20 years ago the idea of genetically engineering our pets was science fiction, and now it's
Right.
so boring that people don't even realize it's happening in the pet store.
Yeah, and I love the idea of the toothbrush. Now I'm always on the lookout for things that give us hope because there's so much polycrisis happening around us. There's so many bad things. There's so many, oh my God, is this ever going to end? And oh my God, what's that barreling towards me? When I see
Yeah,
something like a toothbrush that can do, a genetic analysis for me, that just makes my soul soar. It makes me
yeah,
happy because those are the kinds of things that are going to, give us hope in the future. And I mean, even if it's 10 years down the road, at least it's not 10 years down the road where I'm in a crappier position. It's 10 years down the road where there's actually something in the pipeline that might actually, improve our quality of life. So
yeah.
thanks for
And
that glimmer.
this is always what's driven my obsession with this is I want a taste of what this is going to be like. And I know I spend an hour doing an analysis that I want a toothbrush to do in 10 seconds or something, right? Or I
Right.
send my samples off to be sequenced and I don't get results for 2 weeks. But I'm all trying to look ahead and picture the Star Trek dream, right? The
Right.
toothbrush uses the Star Trek vision,
Mhm.
right? Of
Well,
just pull out the
even with
tricorder.
the plus lives, the metrics, the Qs, the detects, all of those things that we've had that have kind of come and gone, that's felt pretty surreal for me, you know, to go
Yeah,
from, well, we didn't even really do PCR analysis for a really long time to, oh my God, I can have it at my house in a little box,
yep,
what's roughly equivalent. So looking into the future, I think that that's a great perspective and a great way to have, like you said, the Star Trek long view is probably a pretty nice adaptive view to keep you going in a climate where there's so many things that can make us kind of go, why, you know?
yeah. Yeah.
So
We really have come a long way in 5 years, right? I remember
yeah.
when I was first looking at this at the start of the pandemic and just like dreaming, oh, maybe we'll be able to do any kind of testing at home, right? And like it was crazy that like of course antigen tests existed. Most of us, even those of us who were trying to find things, I didn't realize how practical and effective antigen tests were going to be. I thought they were going to be useless because they weren't sensitive enough, right? But they were
Right.
incredibly effective. And then I wrote a post I think it was right around March 2020 or April of 2020. I wrote a post predicting that people would have PCR machines at home. But even then, I was hesitant. I was saying, oh, that they might be $1,000, right? I was not willing to go as far as saying they're going to be disposable $50 tests like the Lucira Check It is,
Right.
right? And
Yeah.
we got that very quickly within a couple of years.
Yeah, that was amazing. It's great, and I'm also really grateful. Like I said, I know that those of us who still live in relative isolation for whatever reason, just having the Plus Life availability or Lucira, whatever, it really opened our world up. It was like, okay, well now I can see my family members. Now I can do X, Y, or Z, and so we're always looking for those kind of, like I said, things or glimmers or ways that things are gonna get better. And so I do agree with you. The demand is going to be an issue in terms of scalability for this. But if we can at some point make it commonplace where people expect to have these things, then the demand goes shoop because it's just expected to be everywhere. And I'll tell you, Rick, healthcare is in trouble in the United States, and
Yep.
probably everywhere. I know that
Yep.
Canada is better in some ways and not better in other ways.
Yep.
And so, having these kind of tools, though, eventually as part of to ease the burden of healthcare. I could really see that. If I've got an easy way where my patient has a toothbrush, they pop out what they have, then they can just tell me, hey, my toothbrush this morning told me that I have strep, you know, then that's a lot easier than having the patient come in and swab or have to go into their urgent care, their PCP, and then wait. We have a rapid assay, but we need a culture and yada, yada, yada.
Exactly.
So I think this actually might be something that is kind of hopeful along those lines as well.
Yeah, to me, it's all about empowering people, right? Taking healthcare. I think we're seeing a lot of things happening in healthcare that are making it possible, and not just possible, necessary, right? There's so much demand for healthcare resources, not enough supply. Increasingly, you got to take
Mhm.
healthcare into your own hands because there's just not enough doctors, not enough experts, or it's too expensive. And whether it's
Exactly.
the technology or AI tools, or I think there's
Mhm.
so many things that are making healthcare more accessible to average people. It's going to be incredibly empowering. And that's certainly how
Yeah.
I feel having this. Right, it feels very empowering to be able to say, what do I have? Where's my viral load at?
Hmm.
Yeah,
Exactly. And then, like you said, then okay, even though my symptoms are resolved, I'm isolating 2 more days to protect the people around me, so I'm not passing it on. So anything
yep,
that's stopping that train of transmission is very important.
yep,
Well, this is, like I said, this is all really super interesting to me, and I can geek out with you for a really long time on it. But I want to go ahead and let people do some Q&A here. So I'll start and see if anyone has typed some like applicable questions in the chat.
mhm.
You did say you do have a master doc of the things that you bought, because there's questions in here about a master doc of reagents and where you get them. Or I've seen a couple of questions here about, is there a starter list for those things that we talked about? Which is why I kind of said, well, let's kind of scale back and go through and go like, what are the basic things in that
Yeah.
in that document? Do you have like the links for where you buy stuff?
I don't think I do, but I'll tell you what. I will post. So first of all, I'll drop into the chat here. I have a website now. I only just created this recently, but let me
Okay.
just start. This is my website. I will commit to writing an article. It sounds like there are people who are interested in getting started. I honestly, when I talk about this stuff, most people just say, "Oh, you're crazy. Like this isn't realistic. Like nobody's going to want to do this." But if like people actually want to try this, first thing I would say is you can just email me, and I'm happy to walk you through it. But
So,
I'm also
Rick,
happy to
Rick,
write an
Rick, Rick,
article.
I'm going to stop you right there because I don't think you understand. We do still have right now in just the still COVIDing community that my ModMin group manages, there's almost 20,000 people. And
Oh wow,
so not everybody is going to want to do this, but
yeah,
there
of
will
course.
be, so let's not just open that up to blanket email
Okay,
just because I don't want you to be like,
sure.
overwhelmed, and then it will be way better. Your website's great. If you can write a blog post with the list, then you can let me know and I can also push it around so everybody
Sure.
has it. And then after, we'll do some caveats, like after you've read Rick's blog post on how to do this, after you've looked at, made sure that people have watched this video, then we can filter down those kind of questions. Maybe we can have you come back and talk about troubleshooting for people who've gotten started. Today we've done sort of a broad overview of kind of what's out there. I think you could come back and talk again about just let's just walk through basic startup. Like here are the 10 things you have to buy, and here's exactly how you would use them just to get started, like I said, with that positive and negative. But please, guys, don't slam Rick with a gazillion emails. Let's centralize them all in one spot. Let's look at his website and get the information there. But Rick, if you have a document, or even if you can do a little starter list with links, even if you want to get it to me so I can push it out to everybody for you, that way we'll centralize information and we'll preserve you as a resource.
Sure.
We
So
would really like to do that.
this is the spreadsheet here. I haven't updated it in years, but this is the spreadsheet that I created when I was getting started. That just lists a bunch of the things I ordered and their prices at the time. Um, and I can do a pass over updating this. There's a bunch of detail in there. So maybe what I'll do is I'm traveling for the next week, but sometime in the next couple of weeks I'll write a blog
Yeah.
post, just a getting started blog post, and then
Mhm.
I can update this with some
That
details
would be
on
great.
exactly what it would be to get
Simple
started.
bare bones version. Here's what you need. You need your swabs, you need your tubes, you need your mini PCRs,
Yep.
like just centrifuge, simple bare bones version for people who kind of want to get started playing with this. In the meantime, people can go to the mini PCR website, look at those different lab kits they have for high schoolers. You can start playing around with that kind of stuff while Rick's getting like the basic list together and stuff like that, if that's something that you're interested in trying. Um,
Yep.
so let's see. Can you talk a little bit about sample prep, which I think you did? Do you have to do the RNA DNA purifications for the rough experiments? We did. We talked about that.
Yeah,
Um,
it's a good question whether you might be able to do crude. I've tried. I have one crude extraction kit. I've tried it a couple times and it's never worked for me. So I haven't put the effort in to try to make crude work. I know it's where a bunch of the challenges with the Plus Life and other testing kits work. So I've always, you know, you know, I've been wanting to get the sensitivity. And so if you're just going to do crude, just use a Plus Life, or the metrics or any of those. It's really only if you want to do that next level of like really getting, it's a little bit more sensitive. I wouldn't say it's worth it the Plus Life does a good job detecting anything that's contagious. So, buying yourself a few more hours at the start of an infection is really not, a big benefit. So I wouldn't worry that much about the greater sensitivity, but
Well, but the question is here, and that's actually the next question. Why would we do this versus Plus Life? Is this just
exactly.
a plan B? And that is really what I thought was worth bringing Rick on and talking about today, because Plus Life, we have access right now. We're working super hard to preserve that access. Sometimes the community works against us in preserving that access, but we're really trying hard to preserve that access. However, number one, and we're going to run into those supply chain bottlenecks probably within the next month or so. And so that's why we told everybody, hey, go get your stock because we're going to run into some of those supply chain bottlenecks. And then, second of all, the whole FDA approval process for Plus Life is still now, it was moving along and then it got jerked all the way back to square one. And so it's probably at least at the bare minimum going to be 2 to 3 years before that would even be a glimmer of a possibility. And in the meantime, if we run into something like people are not being careful and all of a sudden, then we get the whammy down on us again for other reasons, just having the familiarity and knowing if you are depending on Plus Life to open your life, like some people are here, if you are depending on molecular testing to be able to live and move normally and typically in your day to day, then yes, I would say this kind of plan B planning just makes sense. It just is something that you should probably be exploring because we don't know. We hope to always have that Plus Life chain and we hope to be able to keep utilizing that. But if there's something in your life that means that if you lose that, you go back to being basically, isolated, shut in, whatever, even more than you are, then this as a plan B, especially with the mini PCR aspect of it and having the ability to kind of play with it. It's like we are sitting here like Rick was pre-pandemic with his son with the asthma going, oh, I think this might be cool to do. I'm saying, and then when pandemic hit, Rick was like, oh man, I wish I did that. I'm telling you guys now in our community, hey, let's start kind of playing with this and looking at this because we don't want that slam on us because so many of us do rely on the NAAT testing to be able to safely gather, to be able to safely move around our regular kinds of, day to days. So let's please, kind of start educating ourselves. I'm not saying everyone needs to run out and do this because everybody doesn't. And I'm not saying don't buy Plus Life. life right now, because I'm saying yes, if this is not your gig, if you don't love this, if this is not something that makes you go, "Ooh, that's so cool," then right now keep going on with your plus life and keep on trucking. But if you are somebody who has a science background and also depends on plus life, and you do have you've dabbled in this before, or you've thought about it, or you've talked to some. I know I've had conversations. There were a couple of schools that were using lamp setups, which is a little bit different than what Rick is doing. And I think it actually might be a little bit cheaper, but you also have to do it like in a batch kind of setup. It's not, my understanding is it's not great for like single samples. You want to, you
Hmm.
usually have to run plates of more. I could be wrong with that.
So I
Um,
think I did look at doing lamp a little bit. I think the Lucera Checkits were lamp,
mhm,
as far as I know. And one of in particular, they're colorimetric lamp in that rather than relying on something that's fluorescing, they
mhm,
rely on the fact that once there's lots of DNA in there, it changes the pH of the solution. So there's an indicator that turns purple. And you
right.
can pull the checket apart and you can see there's a little camera basically looking to see is this purple or not. You can
Hmm.
actually see the positive tests were purple and the negative ones were not purple. And so the reason I haven't gotten to the lamp, the primers are much more complicated for lamp. So
Right.
they're probably orderable, but it's certainly true that it's simpler. You just need the heater for lamp. You don't need a thermocycler.
Right.
So that certainly
Hmm.
makes it easier. Yeah, and
I'm just kind
there's
of
a few.
sounding the alarm for the people in our community to say, hey, you science nerds who like to do
Yeah,
this stuff anyway and have the capacity, yes, I think we should be looking at plan Bs, whether it's the thermocycler setup that Rick has, or whether we're looking at the lamp stuff. We need to be kind of and then distilling it, right? Because some of us we're going to want to play with it. We're going to play with that micropipette just like you do. Others of us
yeah,
just want to know positive or negative for COVID, you know.
yeah,
And so
exactly.
distilling it for people to get started with just the positive or negative for COVID piece while we're playing with this other stuff, it's just part of our forward thinking, I think, in terms of making sure that our community has access to the resources and the information that we need, regardless of what happens kind of in the chaos that boils around us.
The other thing from a forward-thinking perspective is like we got to make sure companies know there's demand for this, right? Like
Yes.
Plus Life is not a U.S. company.
Mhm.
I know for a fact that there are at least one, probably multiple North American companies considering introducing another one of these type tests to the North American market. So, those companies are trying to get investors. Those investors
But
are looking at signals,
the
right?
problem is that almost every single North American company has folded. And I think
I know.
Q got done. They did Q dirty. The government did Q dirty and sort of forced them out of business, I think. But I think, a lot of the viability in these even metrics, I kind of toggle about whether or not metrics is going to be around for another year or two. I think that I have some doubts there as well. I think actually Plus Life viability, because it's not a US company, might be a little bit even more solid than metrics at the price point and the demand just only inside the US. But I do think Rick's right in terms of finding, letting people know that there is a consumer base that is interested in this, and then just kind of being smart about what's out there. And look, I've seen a couple of other kinds of machines that are kind of on the horizon too. If there's 20 of us looking, or 200 of us looking, or 2,000 of us looking, it makes a difference. And then if we come in and share the knowledge, then we're kind of on the cutting edge when we find something that looks viable and looks also like it would be, useful and practical. Then we can also sound the alarm and say, hey, let's all go get behind this initiative.
The other thing we can do is advocacy to the regulators, right? I think they are overly concerned. I actually find it a little bit paternalistic, right? And I've spoken with the Canadian regulators. I can't speak for the U.S. ones, but when I've spoken with the Canadian regulators, there's a lots of great people there. Don't get me wrong. Great people trying to do the right thing. There's a system that's hard to move politics, but there are some people who have an attitude of like, oh, we can't possibly trust normal people with these things. They might misuse it. What happens if they get what happens if they misuse the device and they get a negative result and then they go and infect a bunch of people? And this is during COVID. I was involved in a bunch of government advocacy and volunteering with Health Canada, and a lot of it came down to trying to get people to compare the options, right? Like, yes, there's risk there, but look at how many people are just walking around not testing,
Right.
walking around with symptoms, right? Like, you got to put it in context of,
Mhm.
and for example, when we were doing the bus thing, everyone was really worried about biohazard, right? And I said, okay, well, let's compare the level of biosafety here to Kleenexes in the garbage, right?
All
Do we
right.
now, when everybody's sick, do we have to start treating everybody's household garbage as biohazard?
Hmm.
because there's COVID virus there, right? And there's like a bit of a disconnect from reality, I think, that just comes from the momentum of this is how we've always regulated healthcare. But look at Europe. Europe doesn't regulate testing devices in the same way. Where's the harm? Where are people dying because they can get plus life? And you can't find evidence of harm as far as I can tell. So I think
Yeah.
there's real opportunity to change the regulatory landscape, but it's
Mhm.
politics. It takes
It
time.
is. It's politics, and change comes sometimes slowly and slower than we want for our families and our ability to kind of go through our day to day.
Yep. And I
There
do, by the way,
is,
I do want to emphasize if you want to get into this and play with this, like please be aware it is still a lot of hands-on time, right? Like it's not very practical. If you want to do the way I do it, I have three separate steps. The sample prep takes me about half an hour and about that's maybe 10, 15 minutes of hands-on time. Then the reverse transcription is just a couple minutes of hands-on time, but it's got to run for 15 minutes or so. And then the PCR step runs for over an hour, and that's at least another 10, 15 minutes of hands-on time. So like, Plus Life is way more practical. When someone is sick in my house, I'll do the PCR thing to figure out what it is, and then I'll test them every day with Plus Life to see, are
right,
you negative yet? Right. Or if
right,
I'm just running out and I'm like, oh, hey, we're about to go to the grandparents' house. I want to make sure none of us have COVID. I use the Plus Life. I didn't use my PCR because
right.
it's
So you're
still,
looking at
it's
two
still a
to
lot of
three
work.
hours on a PCR run from start to
Yeah.
finish, whereas a Plus Life is 35 minutes. So,
Yeah.
here's a good question: are you set up for bird flu? Can you test for bird flu?
So bird flu is just influenza A, I believe. So yes, I think I
Yeah.
haven't actually double checked this, but I believe my influenza A primers would hit on bird flu. I should double check
Okay.
that. It's just
Yeah,
another
it would.
variant of influenza.
Yeah,
Yeah.
it is a variant of influenza A, but you would have to do the actual sequencing if you wanted to know if you had.
Yeah, and so
Yeah.
I've just gotten into. I now have enough influenza A samples in my lab that I finally decided I need to get into influenza sequencing, and so I've
Hmm.
ordered the primers. Influenza is a neat virus because it's a segmented genome. It's got instead of just like a single strand the way COVID is, it's got I think it's 8 different segments, and they reassort and they mix and match in a very
Hmm.
complicated way. But the neat thing is all of those segments have the same start and end, so you can use a single primer that targets all 8 segments. And so I've bought those, I've ordered those primers. I have them sitting in my freezer. I just haven't got around to actually doing the sequencing with my influenza A samples yet.
Well, keep us posted. That could be a whole nother discussion about influenza A and their reassortment and how you figured out how to sequence the bird flu. That'll be really
Yeah.
cool. Alrighty. Well, I'm gonna go ahead and call our Q&A good for right now. We do really appreciate Rick and all of his time. And Rick, I did see in the chat Jessica said, and I agree with her, you are our people.
Yeah. Thank you.
You have found people here that are going to be interested in this and are going to really want to follow you kind of in your journey. So definitely
Great.
keep those blogs coming, and we'll send people over to the website to look for these things. Also, you guys, I am just going to go ahead and hit a share again. If you can and feel inclined and like what we're doing, and you can come in and just like our Kofi, and even come in as a supporter for $1 or $5 a month, then we're going to be pushing out notifications when we find new and cool things that way, and that will just help us keep our team up and running and going. We'll have more insights there coming through Kofi. We are trying to get off of Meta eventually, not immediately, but like in the long term. One of our long term resiliency plans is to not be tied so much to Meta. And remember, we do have the forum, and I've already seen some in the chat about making a nice thread in the forum to kind of follow the people who might be grabbing mini PCR and might be starting to do this. And then that way we can all talk among each other about what we're trying and how we're doing this, and then collectively maybe have a list that we submit to. We email Rick once a month with our collective or something, or something something like that. So, but thank you, Rick, for opening this door for us and letting us being an example of the change that we wish to see in the world, and showing us how it can be done by somebody who's not a lab scientist in their day to day. You're an inspiration, and we really appreciate all the information.
Well, thank you so much, Kara. Thank you, everyone. I'm glad people are interested and curious, and stay safe.